Hi everybody, my name is Hua Tu. I'm the President and CEO of Lake Pharma. Today I will talk about optimizing protein glycosylation and yield with in vitro glycoengineering. On this slide is the introduction, background, and objective about this presentation. Glycosylation affects the pharmacokinetics and function of therapeutic proteins and antibodies, and is an important feature for safety and efficacy profile. Production of therapeutic antibodies and proteins with consistent glycosylation profile has been and remains a considerable challenge to the biopharmaceutical industry. In vitro glycoengineering, also known as IVGE, this technology can modify glycosylation in a controlled manner after proteins are produced and purified, and thus isolate glycosylation management from cell culture manipulations. In this study, we'll examine the efficiency and practicality of using IVGE to control glycosylation and improve product quality at scale. We will also compare IVGE to traditional processes, i.e. cell line modification and process optimization with regard to effects, time, and cost. On slide 3, this is an illustration of IVGE in which enzymes are used to catalyze galactosylation and acylation of proteins. And this content is provided by Roche Custom Biotech, and the reagents we used in the study are also provided by Roche Custom Biotech and are available. Proteins, on the left side, the protein glycosylation, the base module is the G0F. The red is fructose, green circle is mannose, and then the other sugars. G0F has fructose, but then there's no galactose, which is shown in yellow, and sialic acid, which is shown in the purple diamond. But with enzymes, the beta-1,4-galactosyl-transferase can catalyze the addition of galactose at the terminal end of the sugar group. And the second enzyme, the alpha-2,6-sialotransferase, can catalyze the addition of sialic acid, as shown in the diagram. And that will result in G1F, G2F by the first enzyme, and G2S1F, G2S2F, and G1S1F with the second enzyme. And what you see on the G2S, on the right side of the diagram, is that there's different level of galactose, and sialic acid can be added. Note that sialic acid can only be added after the galactose is added. And the enzymatic reaction efficiency will determine the glycosylation profile of the glycoprotein. On slide number 4, outline the experimental approach we take. We did a series of IVGE with increasing scale. At the initial scale, we performed the IVGE protocol at the 2 milligram scale using purified IgG1 molecule. We tested the ratio of substrate versus enzymes, and we also examined the reaction time course to identify the ratio as well as the incubation time that we deem as suitable. Then we move on to the small scale, in this case 100 milligram scale, using both IgG1 and IgG4 molecule. And then we move on to the gram scale and apply the IVGE protocol at the 5 gram plus scale for both IgG1 and G4 molecule. Then we ran through the exercise comparing clone selection and optimization process with the IVGE process, and to see how they compare in the efficacy, time, and cost. Moving on to slide 5, it illustrates the testing workflow. We start with the purified antibody IgG1. The first step reaction is catalyzed using the GalT1, which is the first enzyme to catalyze the in vitro galactosylation. We tested at 1 to 50, 1 to 100, and 1 to 200 and at 2 different incubation time points, 12 hours and 24 hours. After the reaction, we subject the sample to Pro-A purification, then analyzed by mass spec, SE, and CIE F. After the first reaction, we move on to the second reaction, which is catalyzed by ST6 enzyme for the addition of sialic acid. And we tested the substrate to enzyme ratio at 1:10, 1:50, and the reaction time at 12, 24, 36, and 48 hours. And similarly, we clean up the reaction with Pro-A and analyze the product by mass spec, SE, and CIE F. The data is shown on slide number 6, looking at the results of the step 1 reaction, which is adding the galactose group. And what we see is that the original sample is high with G0F, with some G1F and some detectable amount of G2F. But after the reaction, every G0F, G1F, and G2F are all converted to G2F with the 100% addition of galactose. At the 2 different substrate enzyme ratio, we observe that the 1:200, which is a lower ratio of enzyme use, is as effective as the 1:100, and 12-hour time point is really no different from 24-hour time point. And therefore, we use the 1:200 ratio and 12-hour as the future experiment. The results of the second step, dilation, is shown on slide number 7, where we observe the addition of sialic acid, and which is shown in the dark blue and purple bar. The dark blue is G2S1F and purple is G2S2F. The difference is the number of sialic acid group added. And we see that the presence of G1 and G2F is greatly diminished because they're all converted to either G2S1F or G2S2F. Both species have sialic acid content, and we see fairly high efficiency. The 2 ratio used, the 1:50, is about as effective as the 1:10 ratio. So we went forward, we select the 1:50 as the ratio to move forward, and we select the 24 hours as the reaction time point for future experiment. Looking at the sample after IVGE on SE HPLC, specifically we're looking for if there's any aggregation formed during the IVGE process, and we actually did not detect any. So indicating that IVGE does not affect the aggregation profile of the test antibody. So it looks pretty clean and normal. Then we'll move on and at the 100 milligram scale IVGE using both IgG1 and IgG2 as experiment. And instead of testing different ratio and different time point, we fix the first step reaction at 1:200 and 12-hour incubation time, and the second reaction at 1:50 at 24-hour time. After the 2-step reaction, we clean up the reaction and isolate our product using Pro-A and then analyze by SE and mass spec. The glycosylation profile is shown on slide 10 of the mass spec data. Similar to what we observed at the initial scale, we're seeing very efficient conversion to G2S1F and G2S2F. Essentially, after the 2-step reaction, all the product have either 1 sialic acid or 2 sialic acid. And similar pattern was observed using the IgG4 experiment, where the reaction products primarily G2S2F with some G2S1F. And again, in both cases, there's a sialic acid added. SE HPLC showed similar profile on slide number 11. We did not observe an induced aggregation by IVGE reaction. The results summary of the 100 milligram scale is shown on slide number 12, where we observed that IVGE was highly efficient at the 100 milligram scale substrate amount. The step 1 reaction achieved over 90% conversion rate, and step 2 silylation achieved over 90% conversion rate. But they are different degree of S2 formation. We see some sample with 1 sialic acid added, and the second, and others we see 2 sialic acid added. The HPLC results show that the sample do not experience aggregation during the IVGE process. Then we move on to the IVGE at 5 gram scale. We have purified antibody of IgG1 and IgG4, each at 5 gram scale. We performed the same experiment with the same ratio of enzymes and same incubation time as the previous experiment, although here the reaction size is much, much larger given that we have 5 gram material in each reaction. The processes are the same. Results are shown on the slide number 14. The mass spec show efficient IVGE. We've seen the original sample of IgG1 has the highest percentage with G0F, with some G1F and virtually no G2F. But after the first step reaction, we see the major species is G2F, and after the second reaction, the major species G2S1F with some G2S2F. Similar pattern can be observed on G4. The original sample before IVGE is primarily G0F with some G1F, but after the first reaction, essentially all convert to G2F, and after the second reaction, essentially all convert to either G2S1F or G2S2F. So the reactions are fairly efficient at the multi-gram scale. The HPLC results of the samples after IVG is shown on slide number 15, and consistent with our previous results, we do not see changes of aggregation profile before and after IVG reaction. We also tested the sample extensively on the CIE F, looking at the isoelectric point, and what's apparent is that we see a significant shift to the left of PI, so more acidic property after the second step, which adds the sialic acid. And this is consistent with the physical property of addition of sialic acid. What we observe is on the IgG1 case, we observe an average shift of about 1.5 unit to the acidic range, and with IgG4, which is and this particular protein has start with a PI of 7.5, we ended at about 7.1. So there's a left shift. By the way, we have done a lot of IVGE and CIEF experiment. This kind of left shift is pretty consistent and uniform in our experiment. The summary of IVGE at the gram scale is shown on slide number 17. And again, very similar to the initial scale and our 100 milligram scale, the 5 gram scale showed IVGE was highly efficient. And the first reaction and second reaction both can achieve greater than 90% conversion efficiency, but we do see various degree of S2 formation. Now the various degree of S2 formation may have to do with maybe the amount or the condition of the second reaction. And if we titrate the amount of enzyme used in the second reaction, we may be able to drive the reaction to more completion towards one or the other. I want to move on to really look at IVGE to the conventional clone screening approach. At Lake Pharma, we do a lot of stable cell line development, and we often examine the glycosylation profile of clones and use that as a parameter in clone selection. But with IVGE, which is a technology that we can really control and modify the glycosylation profile, it becomes an approach that can really will remove the concern of the glycosylation profile at the clone screening stage. So we went through an exercise looking at several stable cell line development projects of either G1, IgG1 or IgG2, and compare an IVGE approach to a conventional clone screening approach to look at results on product quality, time and effort, as well as cost in achieving that. On slide number 19 is an IgG1 stable cell line production project, and clone screening was performed using two routes, two mutually exclusive routes. The first route has the criteria of looking at the optimal glycan profile as well as productivity, and our second route would only be looking at the productivity, and we would not look at a glycan profile, because we know that IVGE can change the glycan profile after the product is produced. You look at on the table, there's the baseline, which is the pool sample, and we selected 3 clones to examine: clone A, clone B, and clone C. Comparing clone A and clone B, clone A has a productivity of 706 milligram per liter, and clone B has 1,005 milligram per liter. And that's at the original expression, which is in the shake flask. And after we optimized the expression, we could improve clone A to 809 milligram per liter and clone B to 1,447 milligram per liter. And in this case, clone B from a productivity perspective is better. But then if you look at clone A, we do see a higher G1F profile. So normally we might have taken clone A as a molecule to move forward. So that's the A and B, which we're comparing in IVGE. One thing to note that with the glycosylation with IgGs, there's one glycosylation site at the constant region of the IgG, and that site is normally easily glycosylated, but on the other hand, very little salicylic acid is usually detected at that glycosylation site. So just as a note. Now looking at the G4, it's the same thing. We selected clone X and clone Y as a comparison. And clone X has the original expression level of about 1.5 gram per liter. After some process optimization, we improved that to 1.7 gram per liter in shake flask. And for clone Y, which is shown in red, we see about 2.5 gram per liter in the original expression and 3.3 gram expression after the optimization step in shake flask. But if you look at the G1F content, clone X show significantly better G1F content than clone Y. So normally, if we select clone based on glyco profile as well as the productivity, we would have selected clone X, but if we simply look at from a productivity perspective, we would have selected clone Y. So then the X and Y are moved forward to the next experiment. On slide 21, where we examine the clone A and B of IgG1 and clone X and Y of IgG4, either before and after IVGE, and look at the glycan profile, what we see is that there is, again, there's virtually no salicylic acid species with the product of clone A or product clone B before IVGE. But post IVGE, we can see that the G0, G0F, G1F essentially all converted to either G2S1, G2S1, G2S2, G2S1F, or G2S2F. So very different glycosylation species profile. And similarly with G4, for the clone X, the G0F content was 67%, G1F at 33%, and clone Y, the G0 is at 84%, and G1F at 15.9%. Post IVGE, they're all converted to either G2S1F or G2S2F. There's no difference between this, well, there's virtually no difference between the clone X of IgG1 post IVGE and the clone Y of IgG4 post IVGE. So the reaction is very efficient. So now looking at the time and cost comparison of the clone screening approach with the IVGE approach, what we detect here is that if we do clone selection looking at both yield and glycosylation, there's definitely more labor involved, and the timeline is longer. And with because we have to look at glycosylation profile of really each clone, the reagent cost is higher. If we simply look at clone selection based on yield only, it's a shorter time, less labor, and less reagent costs. And IVGE reaction at 5 gram scale, we calculated to be 1 FTE in 2 days with a reagent cost of 27,000. So this makes us believe that a clone selection based on yield only plus the IVGE reaction is a cheaper and faster process than the traditional clone screening approach, and also end with a product that's much higher in the sialic acid content and glyco profile. Slide number 23 is the yield and time comparison of the 2 clones for G1 and the 2 clones for G4. We went through this calculation, and we believe that by the IVG technology allowing us to focus on clone selection using titer and yield as the main parameter, we could actually easily get to clones with a higher yield of the final product. I think the conclusion for this is that IVG could really simplify the clone screening process and shorten the timeline by at least 30%, and effectively increase the yield by quite a lot. Last part of my talk would be on IVGE on non-antibody proteins. The data I just showed you all with IgG1 or IgG4, where the glycosylation is perhaps simpler. So we have been doing IVG on a number of proteins that's non-antibody, where they have multiple glycosylation sites, and the processes are also very different from IgG. In here, we examine how does IVGE work on different residues on different proteins, and then also look at how does the IVGE impact the processes and activities. We can only, at this point, show limited amount of data because a lot of the work are either in progress or cannot be shared at the moment. On slide number 26, here's a target protein that has multiple glycosylation sites, and the protein produced by CHO has a very low sialic acid level. So we use this protein as a test case. We want to look at the sialation level of this by IVGE, and what we did is similar to our initial experiment, where we use 4 different ratio of enzyme versus substrate, and then look at the glycosylation at each of the glycopeptide of this protein. If you look at the glycopeptide A, where we do see sialation just coming out of the CHO cells, we see the presence of G1F, G2F, and G2FSA. And also some portion of G2FSA. But after the IVGE reaction, we see the resulting species are either more clear now. They're either G2FSA or G2FSA2. So with this peptide, the IVGE enriched the assay content and made the species cleaner. Instead of 4 major species, now we're looking at 2 major species. Looking down at the glycopeptide B, we see a significant mixture of glycol species, with the major species as G2SA and G2SA2. And with IVG reaction, there's some changes in that, but not significant changes. With glycopeptide C, the major species is G2F, and after IVG reaction, the major species become G2FSA and G2FSA2. It's a cleaner species. And glycopeptide 4, we also see a more clear of a pattern where G2SA and G2SA2 as the major species. So I think the conclusion we can draw from here is that in each of the 4 glycopeptides we examine of this protein, we actually can see the effect of IVGE, and the degree of sialation before IVGE is quite different, appears to be residue dependent, and after IVGE we can see an enrichment of the sialic acid content I think in every case. So in conclusion of this study, we believe that IVG platform is highly effective at the glycosylation and sialation of proteins and antibodies. And because this is done in vitro after protein is purified, this really enables us to do this all independent of the traditional cell culture methodology in controlling glycosylation. In all cases, the mass spec data confirm the efficient addition of sialic acid, and we do observe an apparent acidic shift of PI on CIEF, and the range of shift is between 0.5 and 2. We're quite pleased with this technology, and we have incorporated IVGE in our process development service offerings. If you want to contact me, my contact information is here. My email is hua.tu@lakepharma.com. My last slide is the acknowledgement. I really like to acknowledge Roche Custom Biotech for developing this technology and make the enzymes and sugar reagents available to the market. The work is done as a collaboration between Lake Pharma Development Center in Hayward, California, and Lake Pharma Gene to Protein Center in San Carlos, California. And the key contributors of this project are shown on the slide 26. Thank you.